It's a great honor to be here, and I have been an attendee of these seminars for quite a long time, and I'm absolutely honored to present our work. I'm going to talk, uh, basically, I'm going to talk about our recent paper in PhysRev Letters, and then I will going to spend as much time as I can about the questions we did not answer and about the interesting things that this paper started us to think about. And I want to start with saying that most of work was done by my great collaborator, Dan Fletcher, Greg Huber, and of course Sid Dansh, who was just a motor of all this research, and he did a lot of simulations, a lot of experiments, and basically I rarely have seen a person so versatile in so different uh, and various things. I will start with saying what is it about. This is about a virus which needs to go through a very difficult landscape, because if a virus tries to infect cells in your epithelium, it needs to go through the layer of so-called mucociliary layer. Here is a video how it works. It's you see that the cells in the epithelium, the ciliary cells near the epithelium always move, and the uh, consequence of this moving is a constant flow of fluid above the cells. And this means that if you have any pathogen or virus or whatever, it will be go with the flow, it will be quickly cleared, and it will be eaten by phagocytes and so on. So basically, this landscape, this very difficult landscape, makes very difficult for virus to infect you. And this leads to a very simple paradox. Here, by the way, here is a simplified picture of what I have shown you. You have epithelium, you have ciliary cells, and you have a flow, and the virus here should go through the flow. Now, if you think about it, the simplest idea you can say is that, okay, I have a diffusion and I can calculate the diffusion of the virus from the layer to the epithelium. And the thing about diffusion is that there is not much freedom for you to play with the parameters. You have Einstein relation, which says that your diffusion coefficient depends only on your mobility and temperature and Boltzmann constant. And you can calculate all this. And when you do it, you get that the time for the virus to move to a cell is really huge. If you plug all the numbers, it's like 20 minutes. It's like a couple of hours. The thing is that mucociliary clearance due to flows is minutes. And this means that the probability for a virion to get to a cell is very low. But cell is not passive either. It tries to do something. So even if you get to the surface, you are not guaranteed to infect. And if you calculate the probability of infection, it's 1/1,000,000. The thing is that the viral loads are much smaller than this. They are in tens of thousands, 100,000 maybe. So if the probability of infection for the given virus is so small, it just basically means that viruses cannot infect us. Basically, it says that Einstein and Einstein equation makes you completely invulnerable to flu, which is nice to know, but this is obviously not what we see every flu season. We have a lot of infections and even with small viral loads. And as I said, that for diffusive particles, you don't have much freedom. You have this and nothing else. So the corollary is that if virus wants to have any chance to infect, it must be active. So virus locomotion must produce and dissipate free energy. And this is a huge paradox because, okay, basically, what does it mean active? You must have some energy stored, you must have energy tank with you, you must have motors to do, and virus is small. It doesn't have any space for all this. And what happens is that virus is very smart to do it. Here is a model, and basically the model is that virus has some binding proteins which bind to filaments in the mucociliary layer. You can think about your mucociliary layer as a lots of small filaments in a fluid, and virus can bind. The binding itself is reversible. It means that you cannot have any active things there, and Einstein still rules. But besides reversible binding, there are some other proteins which break the bonds, but it's important they break it irreversible. If you basically they burn the filament, and if you burn the filament, you cannot bond again. Because you cannot bond again, you change the entropy of the system, you change the free energy, and you have free energy production. Here are some movies from the paper by Fletcher and Wahi. I hope you can see it. And basically, we put some, they put some cleavers and binders, and as you see, you have some sort of a fast active motion. You have some sort of a protein rockets which move around. Just a moment. What I want is to use this. Just a moment. Okay, here is our model. And in our paper, we discussed one-dimensional model. The model is very simple. You have uh, receptors, you have filaments, and filaments have receptors. Receptors could bind and unbind, and you have the constant for binding. And there are some burners which can burn the receptors, and you have some constant for the rate of burning. When you have this, you can get the energy balance, and the energy balance very simple. You can calculate the energy, free energy production by cleaving, and it dissipates into the virus friction. Another thing which is important, but it's a little bit less evident, is the friction of filaments. Basically, when virus starts to move, when you break the filament, if you think about it as a spring, the spring goes back, and because it goes back, there is some additional friction and additional production of heat. So basically, you write down a very simple equation, it's conceptually simple. I want to demonstrate the main equations in the paper itself. Basically, you say that your energy depends on the probability of site of filament to be burned. You can calculate the probability of this filament using the equation of motion of the virus. You can calculate the friction of the virus, which is just a simple hydrodynamic things. You have something which is proportional to the square of the velocity. And it's a little bit more involved, but possible to calculate the probability, the friction by filaments. You put this into equation like this, and this is a closed form equation for velocity, and you can calculate the velocity of the virus. So basically in this model, virus just moves with a constant velocity, which depends on the cleaving constant, and on the burning constant, and of course on the friction. The most, okay, here are the results. This is the results for the virus where cleaving and burning sites are mixed. So basically this is a symmetric virus. You would ask why symmetric virus would move at all, because both moving to the left and to the right is the same. And the answer is that we found that there is a spontaneous symmetry breaking. If you start to move in some direction, you start to burn mucociliar layer, and you cannot go back because this mucociliar layer is already burned. So you want to go further and further and further. So there is a symmetry breaking. Of course, if you have asymmetric virus from the beginning, it's even faster and it moves even faster. And the interesting thing is that there is a sort of a Goldilocks zone. Here is KD uh, uh, is a binding constant, Kc is a burning constant, and what you see is that if you bind too much, you are stuck. If you don't bind at all, you're stuck because you are basically diffusive particle. And the same goes with the cleaving constant. If you cleave too much, you burn all around you and you are stuck. If you don't burn anything, then you don't have active motion and you are stuck. So basically you have a Goldilocks zone. And this Goldilocks zone actually depends very critically on your environment. If you change your mucociliary layer, if you change chemistry of the filaments, if you change the density of filaments, you get different optimality constant. And basically, if you move fast inside human mucociliary layer, it means that you probably would not move fast inside avian or swine mucociliary layer. And this means, first, this means a very interesting thing. We found out that the real viruses are selected for this. We found that the parameters of real viruses are very close to optimal parameters which give you the maximal speed and maximal chance to infect. But because the parameters are different for different environments, it means that if you have, for example, bird flu, and bird flu wants to jump to humans, it must mutate. It must change its parameters so it can now move in mucociliary layer. And so what we think now is that one of the most important mutation, probably there are other mutations, but one of the most important mutations for the virus to jump between species is to change the constants of burning and binding and make it better for this environment. Another thing is that we understood why Tamiflu works. Basically, what Tamiflu does, it changes the burning constant for the virus. And because it changes, you go outside of your optimal zone and you start to, the virus starts to move slower and infects worse. But also we understood why Tamiflu works not very efficiently, because basically you try to move in this direction. And you see that for asymmetric virus, changing of Kc, changing of the burning constant is not very critical. It looks like changing of the binding constant would be more critical for the virus. And one of the conclusions of this paper is that we probably need to look at the antiviral drugs which would change the binding between mucociliary filaments and virus proteins instead of changing the rate of burning mucociliary filaments. Okay, that's basically what we did in this paper. And now the questions we have. And first question is technical, because we wanted to make a very simple model. We did a one-dimensional model. We had a lot of two-dimensional models. But what happens in 2 plus epsilon dimension, basically you have a mucociliary layer which is thin but not of zero thickness. What happens of 3D? Basically, you would say that, okay, I have ballistic motion at small times, which becomes diffusion motion with new constant large times. How fast? And it would be very interesting to develop a theory and develop a think about this. Second, gradient and virus motion. The thing is that if you change the density of filaments, you can show that virus moves against the gradient. It tries to move to the place where there is more sialic acid. But what does it mean against the gradient? There is more sialic acid where the epithelium is. So basically, it means that viruses not just move actively, they actually move actively towards epithelium. So they even are more smart and better to infected cells. And it would be very interesting to describe it and have a good theory and simulation for this. The third thing, we started to think about this. When you burn your sialic acid around you, you basically change your environment, right? And we started to think about random walker. The classic random walker just moves here and there, and we know how it works. There are some things as self-avoiding random walks. Basically, you are not allowed to return to the place you started with. And what the virus suggests, that there might be a third possibility, where you move around, you don't like to return to the place you started from, but this is not a hard uh, thing. You still can do it if there is nothing else, because you basically you can return to the place using your diffusion motion or the rest of sialic acid. So we have this self-occluding walk or sower, and this is, by the way, one of the most famous sowers we have seen. And the question is, what do we know about long-term asymptotics of self-occluding walks? What do we know about this situation? Would it work? Obviously, in 3D, it would be probably like random walks, because you don't return very often to this place. But what happens in 2D? And how the statistics of these walks works. And it's very interesting because it will give you some estimate for virus motion and for the time the virus goes to do. So basically, what we understand, and here are our conclusions. The first thing, that life is an active process. It's a reversible active process. You need to burn you you need to burn fuel to live. And life is cell machines which are irreversible and active. Now we understood that flu viruses like do what viruses love to do. Instead of using their own machines, they basically steal somebody else's cell machines. And we know that for the virus replication, they steal your cell machines and use your cell for replications. What we did not know, and what we know, that they do the same for motion. They steal the energy that cells expend to create mucociliary layer and use it for their own locomotion. So again, viruses do what viruses love to do, steal something. And what we found is that viruses are selected for this process. Basically, it means that their parameters are such that this motion is fast. And because we now understand the strategy, we have another tool to predict and prevent the motion of viruses. So that's what the gist of my talk, and I'm glad that I still have several minutes for questions.